Good morning, everyone. Thanks to the Soil Health Institute for inviting me, and I'm extremely honored to be included with such a distinguished panel here. So I'm going to continue on with the idea of harnessing the phytobiome to build disease-suppressive soils, and I don't think it's too controversial to say that when it comes to soil biology, beyond measuring abundance, we really don't know what we should measure at this point. We know communities are changing, we know some of the functions, but how do we move forward and come up with a good index of the properties of that soil? And in this case, we'll be talking about disease. So I think everyone in this room can probably come up with a similar definition here of soil health, but the key is that it's three components. Physical, chemical, and biological processes or indicators all interacting to drive soil health. And second to that, it's a living, functioning system. This cartoon here is really just to document that the living part is an interplay of both beneficial and detrimental or pathogenic organisms, resulting in the function of that soil. Okay, so relating this to disease, most of you or all of you, I'm sure, are familiar with the disease triangle, where disease is now an interaction of pathogen, host, and environment. I'm going to ignore climate here for a second, which might offend some, but I replace environment with soil. And so what we're talking about here is, in terms of disease, is how that soil health or soil is interacting with the pathogen and host and affecting disease. With these three different processes, biological, physical, and chemical, they're all going to have different impacts on plant nutrition, plant defense, induced systemic resistance, acquired resistance, and pathogen growth. And we could argue about the relative contribution of these different processes, but I think just want to make the case, in my mind, biological is one of the key factors influencing all of those and the development of disease. Hopefully I convinced you that we should be looking at the biological component. So how do we measure that soil biological contribution to disease suppressivity or the soil suppressivity? Oops, and I went the wrong way. Red arrow this time. Okay, when we look at the microbial community, there are different levels we can measure. We can start out and typically, and the easiest is to measure abundance, how many microbes are there, things like microbial biomass, carbon, nitrogen. We use PLFAs or DNA a lot. But that just doesn't give you an idea of who's there or what they're doing. So we can move down to things like composition. And so this is which way to point. Fancy little tree in there of just documenting a taxa in a soil. That gives us an idea of who's there, but beyond that, names are always changing, their functions might be changing, so we need to frequently move beyond that and understand what those microbes are doing. We can look at things like enzyme activity or respiration or gene expression. But the big million-dollar question here is there a cost-effective measure or index that incorporates all of these components and is useful for predicting the status of a soil or its ability to suppress disease in this case? In our laboratory, we typically use next-generation sequencing or high-throughput sequencing. And we use this method to look at, we start taxonomic specific abundance. We can look at things like species richness or diversity. But what I really want to talk to you about today is this idea of looking at specific genes or beneficial genes associated with different soil properties. And so I mentioned the idea of being cost effective. So the concept here is one assay And we look at multiple genes related to different benefits of the soil. And we're using a 16S gene, which is probably has the most information about taxa. And we map that to function. So here's sort of the typical process. As you all know, everything has DNA. So we start out with a soil sample, we extract DNA, and then we need to amplify a specific gene. In this case, we're looking at the 16S gene, so if we sequence it, typically we have an idea of what taxonomic group that microbe came from. From there, we identified an abundance based off of that PCR of the total community, and after that, sequencing, and we can identify a taxonomic specific abundance in your soil. Moving from that, we build a gene index. So we take advantage of whole genomes that have been sequenced, and we map the different microbes to the genomes, and we end up with our taxonomic tree. We map to different reference genomes, and then we end up with gene-specific abundance. So this is the idea here. If most bacteria microbes or bacteria have 4,000 genes in them on average, what genes are we going to target here to understand soils? So put together a list here of genes that all relate to different processes or beneficial processes in soil. Everything from carbon decomposition, nitrogen fixation, or down to siderophore production, which we'll talk about in a little bit as that relates to disease. A couple of examples here where we've used this index just to show you differences that it's influences by plant cultivar. So in the top figure, these are all different potato varieties being grown in a common garden, part of the Western regional trials in California, Colorado, and Texas. You can see each cultivar has different abundance of these different genes. And I'm not going to go through the different genes, but there are differences, and it's related to that plant variety or cultivar. And then down here in this figure, again, this is a wheat trial, and these are different wheat varieties. So we're picking up not only differences between the two different species, but we're picking up differences between the different cultivars. And we've done a series of work with the Western regional trials, and what we're seeing, the microbes associated in the index of that gene relates to things like nitrogen use efficiency or disease, but we'll leave that for another day. Another example where we've used this index, this is a study in eastern Colorado looking at cropping intensity. So basically just starting with a wheat fallow, so fallow every other year, and then wheat corn fallow every third year, all the way till we get to a CRP or perennial grassland. And again, this index picks up a nice difference between beneficial bacteria. So as you basically remove fallow, we're increasing the microbial biomass, we're increasing the beneficial microbes. So the index is very sensitive to management as well. Okay, so we can see differences in this index, but can it predict soil health and plant function under stress? In this case, disease, suppressivity. So we worked in the San Luis Valley of Colorado looking at different cover crops and how they might reduce. Initially, it was a study to see how they reduce nematodes. But we also, when I was invited to work, we started to look at the whole microbial community and how cover crops are affecting that community. So this, I think, is a really exciting picture here. So this is the tuber yield. On the left-hand side here is a fallow treatment, and on the right-hand side is a sorghum Sudan, or Sudan 79. And you can see that not only did it increase the number of tubers, but we have a higher quality tuber. We tend to have more marketable tubers over 8 ounces. And I think so our estimates on that study was that the sorghum 79 was increasing income anywhere from 60 to 400 dollars per acre. My colleague Jorge Delgado has done a lot of work looking at nutrient use efficiency in these plots, and that's part of increased nutrient use efficiency is part of what's driving this yield increase. Okay, so now let's just step back a little bit. I mentioned genes and what genes we might look at, trying to relate that increase in nutrient use efficiency with the microbial community. Siderophores. Siderophores are a compound produced by bacteria, plants also, but they result in the chelation of cations, iron, magnesium, and so on. And once they're chelated, that helps both microbial and plant uptake, so that which can increase your nutrient use efficiency. They are also implicated or have a role in disease suppression, so that chelation can basically rob cations from either your pathogen or fungi, so it's a mode of antagonism. And here this is a really simplified schematic of the pathway for siderophore production, but there's essentially 3 key genes. So we've targeted these 3 key genes, and those are part of our index. Okay, so told you when I started on this project, we were trying to relate this to microbial communities with the different cover crops. So we have our fallow, canola, honey sweet, that's a sorghum Sudan, mustard, and the Sudan 79. You can see it so the top figure shows the increase in microbial biomass across essentially all of the cover crops, a significant increase in microbial biomass. But what is it doing? Well, we also looked at just soil activity. So this is a colorimetric assay for siderophore activity. And we see a similar pattern that the siderophore activity is increasing with cover crops. Well, what does this mean? As I mentioned, siderophore activity has a role in disease suppression, but we also, we'll step back a bit, we isolated microbes from each of those soils. And in these soils, we assessed the microbe's ability to reduce rhizoctonia, as well as 2 other potato pathogens. So this is just sort of our bioassay of microbes. But what we saw was in the sorghum Sudan, where we saw the higher yield, we see more siderophore activity. We're also seeing more biocontrol activity. We see a very high diversity of isolates, but we can only grow a limited number of isolates. So the real question is, what is in those soils that's contributing to biocontrol? This is from sequencing data of this entire study, where we look at each node in this circle is a different genus of bacteria. And it's all the genera that we found in the study that have the first 3 genes here are those siderophore genes, and the rest are other different genes implicated in biocontrol. So one of the key things is now when we do a sequencing approach, we see, I think, I added this up once, 17 different phyla now that all have biocontrol activity. And I don't know about anybody else, but when we normally think of a function with microbes, whether it's nitrogen fixation or disease control, or normally there's one or two organisms that we talk about first. If it's nitrogen fixation, it's rhizobium, or if it's disease control, it's streptomyces or bacillus. But normally not such a wide range of microbes. So these functions are the key here is that these functions are redundant, so these circles show whether that function is in that group. Functions are not only redundant within a group, but they're distributed across a wide range of microbes. So we need some sort of way to estimate that entire population that's contributing to this function. Okay, with that in mind, so now we see that there's a great diversity. We identified a few genes that we can look at. Can we now predict what that soil health or function is under stress? So we took soils from these different cover crops, basically made an inoculum from those soils and applied it to potato plants grown in a sterile media. And from there, we inoculated with rhizoctonia, and we monitored disease development. This graph here shows you the disease development. So a 1 is no disease. I don't know if anyone can read the legend down here, but basically no disease, so a negative control. If we didn't inoculate, we didn't get disease. If we inoculated, we got a high level of disease. And then as we added the inoculum from the different soils, we see a decrease in disease, and our Sordan 79 had the best disease control. So I said part of the goal was to measure a soil and come up with an index and predict what its biological activity was. In this case, in the top graph, this is the percent of the community or that has the siderophore activity. And so here's the fallow, has the lowest amount, Sordan 79 the highest, and this is one of the 3 genes in that pathway that the ENT-A, but it's the most common of the 3. And each different color in this is it's colored by genus. So you can see that even within fallow, we have, I forget how many, 50 or so genera that all have this activity. And then Sordan, we have 50 or so more, but they're more abundant. So down in the bottom here is relates that index, and I'm almost out of time, relates that index to the actual soil activity for the siderophore production, and we see a nice correlation. So we've predicted that function or that biological activity, and I'll end with just saying this is similar where this is that same index of the biological community or the siderophore community versus disease, and we have, again, a nice relationship, and we've predicted that disease index. Some of the benefits of this approach, we've just talked about siderophore here, or disease, but with a single assay, we can provide an index of many different functions depending on which gene we look at. There's some things to talk about here about what source material you use, whether it's DNA or RNA. They both have pros and cons. DNA a little more stable, but it's not quite as variable either due to weather or it's easier to handle the samples. But the idea is that this index is sensitive to management. We talked about cover crops, we talked about cultivars. We've also done a series of studies with tillage. And then last, I just want to put up here, we've been working hard on a software tool that allows you to analyze this complex data, the sequence data, and the information's here on that, and it will not only analyze your sequence data, but it'll give you your index of these different genes. And I'm out of time, so thank you.